Now, I think this title that was given to me deserves a couple of comments. So muscle memory has traditionally been used more or less synonymous with motor learning. So it is this concept that if you have learned a motor task, riding your bike, for example, even if you stop riding for a couple of decades, you can easily reacquire that skill. But it has also been used, and there is this for strength tasks, and there is this training studio folklore, at least, where the old guys would be sitting saying that, well, you know, I was strong, I did strength training when I was young, and I haven't trained for a number of years, but it comes back so easily to me. And for such motor tasks, I don't think motor learning in the CNS has been a satisfactory explanation, because in order to have strong muscles, you also need to build muscle protein. So I'm suggesting, I'm going to submit to you that there is a muscle memory residing in the muscle cells themselves. This has been considered a pseudoscientific concept, this idea of a body memory, but of course there are other examples as well, like in the immune system, where you also have forms of cellular memory outside the brain and the spinal cord. But before we do that, we have to get into Sherwood Forest, the Sherwood Forest of muscle fibers, the muscle cell, which is a rather peculiar cell. It is by far the largest cell in the mammalian body, much larger than the egg, for example, and it has cytoplasmic volumes several orders of magnitude larger than the typical spherical cell. And the way, in my opinion, that muscle cells deal with this vast volume is by being multinuclear. And in fact, it's one of less than a handful of syncytial cells in the mammalian body. And the idea has been, and this is an old idea, it goes back to the 19th century, that each nucleus supports a certain part of the cell or a certain amount of cytoplasm, a myonuclear domain. And in its strictest form, this hypothesis says that the myonuclear domain is constant. That is that each nucleus can only support a certain volume of cytoplasm. And it, of course, then a larger fiber would need more myonuclei than a smaller fiber. If you double the size of the fiber, you would need to double the number of myonuclei. And going with this too, also, if you change the cell size, that is hypertrophy or atrophy, you would need to change the number of nuclei in order to keep the myonuclear domain volume constant. Now, the model, and I think most here will agree that this has been more or less the textbook model for muscle size regulation, has been that when you have hypertrophy, you start growing your fiber, and then you add a nuclei from fusing satellite cells, and you end up with a larger fiber with a higher number of nuclei. And then if you go back down again, or if you go down again, an atrophy condition, then you lose these extra nuclei by apoptosis. This would have to be a selective apoptosis within an intact cell, and that would have been a rather interesting mechanism. But I should say that this has now become controversial, and I would say that this model is in many respects flawed. And I'll come back to that. And then you might ask, why is there such disagreements in the field? And I think one major reason for that is that it is very hard to identify myonuclei at the light microscopy level, and a lot of the literature is based on conventional H&E staining. It has improved somewhat with immunohistochemistry, but I still think it is very hard to separate myonuclei from other nuclei of the tissue. I should remind you that about half of the nuclei in the muscle tissue are non-myonuclei. So a few of them are satellite cells, but then there are all other kinds of stroma cells connective tissue, vascular tissue, things like that. So if you are a biochemist or molecular biologist and just grind up your muscle, like many of us has done, then this could be a confounding factor, obviously. It's a lot of other things than muscle nuclei inside the tissue. Now, I think it is hard to identify myonuclei at the light microscopy level. I think it is a dangerous exercise. I'm not saying it's impossible, but unless you use a very strict definition for what is a myonucleus, you will get variable numbers of myonuclei. For example, we define a myonucleus as a nucleus that has its geometrical center inside the inner rim of the dystrophin staining ring that you'll get on a cross-section like here. If you switch that to the outer rim, you will get a higher number of myonuclei or a higher fraction of myonuclei. So obviously, this is very sensitive to how you define your myonucleus, and I think that might have created confusions. So in my lab, we also use histology, but our gold standard is in vivo imaging of myonuclei, and it's in principle it's very simple. We put an animal, a mouse in most cases, under the microscope, open up and inject then a nuclear dye into the fibers. In this case, it's an oligonucleotide, but you can also use expression vectors for reporter genes, and you can inject then a number of fibers, and you would know then that the nuclei you observe here are the nuclei that is confined by this particular sarcolemma, which is then your myonuclear for that particular fiber. And I'll just mention a few observations we made then with this type of technique, a few basic observations. So first of all, these nuclei are fixed in space over time. This is different from tissue culture, where the nuclei are swimming around quite rapidly. But they are fixed in space over time. So this is day 0. We come back 4 days later looking at the same fiber, and you can see that the relative position of these nuclei has not changed much. The other feature I would like to point out to you is that the distribution is not random, it's rather irregular. And we have dealt with this with computer simulation. So this is the position of a real fiber, the nuclei of a real fiber. If you take the same nuclei and distribute them randomly, it will look more like this, and you see it's quite different. Some nuclei are very close together. You rarely see that in vivo. Now, what would be an optimal distribution? If you want to optimize the distribution in order to minimize transport distances to all parts of the cytosol, what would you do? Well, paradoxically, that would be to place the nuclei as far from each other as possible. It's like heating up a room. You distribute your space heaters evenly far from each other in that room. And this is then such a placement. It's a computer algorithm, a repellent algorithm. And of course, it really looks like this. But if you add a little scatter to that optimized distribution, you will end up with, for example, a pattern like this, which looks very much like what you observe in real life. So there is an optimization of position of the nuclei. And I should say that this is reduced in the elderly. They are less ordered in 23-year-old mice. And so that could contribute to this phenomenon of muscle weakness in the elderly, sarcopenia. I think the ordering of nuclei in the muscle fiber is of functional importance. This is a paper in Nature from Bailey's group where they studied the Drosophila larva, and by knocking out MAP7, so also the nuclei of the Drosophila larva is ordered, but they were able to make them disordered by knocking out MAP7. And as you can see, you then get even a clustering maybe of nuclei, and they show that this insult, this change, led to a decreased ability to crawl for these larvae, so impaired muscle function. So I think we should look at the disorganization of myonuclei in many myopathies, not only as a symptom, not only as a sign, but it could also be a causal factor. Now, how about myonuclear domain? So are they really constant? So this is now measurements from fibers in the soleus muscle. That's a slow oxidative muscle. And if the myonuclear domain is constant, then the observation should follow this line. And it does in a fairly good fashion. So you might say that there is a case here for a more or less constant myonuclear domain volume. If you look at the DDL muscle, which is a fast glycolytic muscle, then there is also here a correlation between the size of the fiber and the number of myonuclei, but it is less steep. So in this case, it seems to follow a law, if you like, saying that the number of nuclei increases proportional to the surface area of the fiber rather than the volume. So there is then an area domain that seems to be constant. But this is by no means universal. If you look at middle-aged mice, these were fairly young adults, the previous ones, this would be 14 months. There is really no correlation with this type of technology between size and the number of nuclei. And I think this might be related to my hypothesis that the number of myonuclei in the fiber is not only reflecting the current size of the fiber, but also its previous size. In other words, there is a memory phenomenon here, and I'll come back to that. Now, as I said, we were very interested in this part of the hypothesis. So is there really an elimination of, and we were actually we didn't ask that question. We asked how can we study that in more detail, the elimination of nuclei. That was our original idea, by apoptosis during atrophy. But to our great surprise at the time, we observed no elimination of myonuclei during atrophy. So this is an example of a fiber at day 7, at day 0, I mean, and it was then denervated at that time, leading to atrophy, and then 21 days later, we came back and looked at the same segment. You can see the neuromuscular end plate here as a marker, and I think you can count that there is 21 nuclei in this segment here, and there is still 21 after 3 weeks, irrespective of a rather pronounced atrophy. And we did this in a number of fibers, and the result is the same. There is no loss of myonuclei. We had a hard time publishing this since it went against, as one referee said, a large body of well-performed literature. So we had to do it in several atrophy conditions. So we had done denervation, we have done TTX impulse block, we had done antagonist ablation, we had done hind limb suspension, and we had done detraining. And in all cases, we see an increase in the number of tunnel-positive cells. There is certainly apoptosis going on in the muscle, and it is increased during these conditions, but it's just confined to the nuclei that are not inside the muscle fiber. We only see it in nuclei outside this dystrophin ring. Okay, so in my opinion, this part of the model is not correct. And since the rest of the model also is based on technology that might have been imperfect, we also wanted to reinvestigate that part. So is there really a fusion of satellite cells going on during growth? And the answer to that, to our relief in a sense, is yes. So you will see when you overload, this is synergist ablation model, but it's on the EDL. And when you overload the muscle, you see that there is an increase in the number of nuclei, and then following that in time, there is an increase in size. So this was opposite to previous literature, but we confirmed that by seeing that there is a temporary a decrease in the cytoplasmic domain volume corresponding to the nuclei coming before the growth, hinting, of course, that there might be a causal relationship between incorporating nuclei and growth. So I think that most people in the field will say that under at least many hypertrophy conditions, yes, this happens. Hypertrophy is accompanied by fusing of satellite cells. It's accompanied by an increase in the number of nuclei, at least under many conditions. But then you just heard Charlotte, and she is suggesting that there is sort of a shortcut here that you can skip this step. She is saying this is not obligatory. You can have a growth in fiber size going from a thin fiber with few nuclei to a thick fiber with still few nuclei. So in other words, this is obligatory. And my answer is, unfortunately, I should say, the opposite of Charlotte's. I think it is obligatory. And it is even based on exactly the same type of experiment she described. We used the same mouse model where we ablate satellite cells that she described elegantly to you, so I'm not going to go over that again. But in these mice, where there is perhaps only 10% of the satellite cells left, we don't see any hypertrophy. So we did, this is the plantaris, the same muscle as they used. We then observe in the control group a 26% hypertrophy when we overload. This is completely extinguished when the satellite cells are not present. And I think it is important, I'll come back to that in a second, that we also did this in another muscle. It's an EDL muscle. It's also a fast muscle, as fast as the plantaris, I think, and the same. Basically, the result was the same. Satellite cell seems to be required for overload hypertrophy. And I think in this case, the result was particularly clear, since if you look at the distribution of cross-sectional areas, it is the same in all groups, except when you combine the presence of satellite cells with overload, where you get, as Charlotte said, this characteristic shift to the right. It's a little bit more fussy, I should say, in the plantaris, but also there it's pretty clear that the only condition that really gives you a right shift is the combination of satellite cells and overload. And then, so why are the results so different? And I'm going to give my opinion on that. Charlotte might have a different opinion, but I'm going to give my view on that. So this should be not 13, this should be McCarthy et al. 2011. And I think this is, and they are very clear in their conclusions. They think that you require satellite cells. And my sort of explanation is that this plantaris overload model has its problem. It's very harsh. So you ablate the gastrocnemius, and that's a very large muscle, and the soleus, and then the plantaris is left to take all the load, and the plantaris is a relatively small muscle. Secondly, the blood supply to the plantaris comes through the gastrocnemius muscle that you are ablating, so you can easily damage blood supply. And in that paper, McCarthy 2011, they report that there was 30% of fibers that showed signs of damage and regeneration. And those were, as far as I understand, included in their analysis. We saw less of that, and we saw almost nothing in the EDL muscle, and we did not include such fibers in our analysis. Also, I think this regeneration problem might have contributed to a rather small hypertrophy. So we have tried to calculate that from their figures, and to us it looks like the hypertrophy in the control group was only around 10%, and that's a very small scope to test the importance of satellite cells. So I would rely more here on the EDL model, that I think is more gentle and where you have less problems with the impairment of blood supply. Now, I wish that Charlotte was right, because there is a conceptual problem with our model. So if you get nuclei each time you go up and never lose them when you go down, can you then pump up your muscle by sort of exercising, detraining, exercising, detraining? I mean, I always used to get that question when I gave these talks before we had the results I'm going to show to you now. But the answer is no. You cannot pump up the number of myonuclei in this way. And that is shown by the following experiment. And this is a case where you have radial growth without adding satellite cells. So what we did then, together with Dr. Deplanche in Lyon, was to use this hind limb suspension model. So we had a normal state, and we lifted up the tail, and then you get atrophy in the hind limb muscles. And we observed then that you got the thin muscle fiber without losing the nuclei. And then when we let the animal back down again, then the fibers grew back to where they started, and this rather dramatic radial growth did not involve an increase in the number of nuclei. So they remained constant throughout the experiment. And Charlotte's group has shown directly later the same year, publication, that this can happen, this regrowth can happen with the satellite cells ablated. So I don't think that you see radial growth in this case because a high number of nuclei are not needed, but just because they're already there. So what is then determining the number of nuclei you see in the muscle fiber? Well, I'm suggesting that what you could call the peak pegging hypothesis, inspired by this thermometer, which is such that the mercury column will push up this little peg and leave it at its highest temperature that's ever had. So it's possible that the number of nuclei you see in a muscle fiber reflects the largest size the muscle fiber has ever had. So if you don't go beyond that, you will not recruit satellite cells, you will not get an increase in the number of myonuclei. If you go beyond that, you might need to add more nuclei. And I think such a view will explain some of the findings in the literature discussing different kinds of roofs for recruiting or thresholds for recruiting satellite cells. So I'm going to talk about muscle memory and doping in a second, but we are suggesting that perhaps another model should be used than the one I started with, and that is that there is a first training route where you take untrained fibers, and then satellite cells are required for hypertrophy. Then you detrain, you don't lose the nuclei, and then there is a retraining route that you can take skipping this step. And then, of course, is there a benefit to this retraining route? Or is it just a strange phenomenon? Do this retraining come easier or faster than taking this route? So in order to test this, we used testosterone, and that was not because we were interested in doping, but that seems to be a very interesting side effect of the paper. But because it is reversible by hypertrophy model. And so we used female mice, and you can see that there was a tremendous increase in cross-sectional area when you gave steroids. It was actually stronger than by overload alone, and the effects of the two were additive. And we showed this both in the Soleus and in the EDL. Now, this was done by implanting a pellet under the skin, a testosterone pellet, and you see that then the testosterone goes very high, but it goes back to immeasurable levels within a couple of weeks. Actually, the pellet was actually removed at this stage, so there is no further testosterone given to the animal. And then you can see that when we measure then 3 weeks later, the muscles in terms of size were indistinguishable. So this doping episode, if you like, the effect on muscle size, muscle fiber size, had been completely removed. But what lingered was the number of nuclei that remained higher in the testosterone group compared to the sham group. And then we waited, I'm not showing you all the experiments, but in this experiment we waited then 3 months. So there was a 3-month washout period with no testosterone. That's a very long time for a mouse. It's about 15% of its lifespan. And then, of course, at this stage also, there was no difference in size between the two groups. The cross-sectional area of the testosterone group and the other group was exactly the same, but the number of nuclei was still elevated. And then we started overloading. So what do you think happened? Well, the group that had been doped, if you like, grew 36% within the first 6 days, while the control group grew insignificantly 6%. So this is an effect of the steroid. The steroid is long gone, cannot be measured. There is no gene that is altered. There could be an epigenetic mechanism, I guess, but so it would be extremely hard to show that these animals had what these had gone through, except that they have a high number of nuclei. So I see that you are getting impatient, and clearly this caught a lot of attention for the implications for doping. And Justin Gatling said that for the few haters out there, when he was confronted with these findings, you know his story, right? He has been caught 2 times for doping. He's Olympic gold medal winner in short distance running. For the few haters out there, it seems like that's what they want to do, discredit my name and label me with the laboratory rats in Oslo, as in The Guardian. Also, there was a lot of press on this, and Science News selected 25 Science News of the year 2013. Top stories, 25 top stories, everything from meteorites and to biology. And we were among those 25. I had to admit we were number 24, but still we think that was a lot of fun. I will just mention one more thing, and that is how long do muscle nuclei last? How long do myonuclear last? And there's very little literature on that, but there is a study measuring isotopes from nuclear bomb testing in the post-war era. And the conclusion there is that muscle tissue will have a half-life, the nuclei of muscle tissue will have a half-life of 15 years. I think that the myonuclei might be more stable than that because there are other cells that probably has a higher turnover in the muscle that would tend to reduce the number of years. But at least 15 years would be the half-life for myonuclei according to these studies. And that brings me maybe to a more positive note than doping, and that is that I think this could have implications for healthy aging. You should know that muscle weakness, sort of the physical Alzheimer, it's a big problem in the elderly, and it is much harder to get these satellite cells into the fibers when you are old than when you are young. So my advice, and advice maybe to the population, should be to start early. Thank you.